Laboratory directions for lymphatic organs. Learning objectives for lymphatic organs. 1. Be able to identify and describe the histological detail of a tonsil and distinguish between tonsils. 2. Be able to identify and describe the histological architecture of a lymph node. Be able to trace both the flow of lymph and blood through a lymph node. 3. Be able to identify and describe the histological architecture of the spleen. Be able to trace the blood through the structure of the spleen and relate this flow to spleen function. And 4. Be able to identify and describe the histological architecture of the thymus. Be able to relate the structure of this lymphatic organ to its function. Scattered throughout the body are the various lymphatic elements that form part of the body's defense system. Generally distributed so that noxious agents entering the body soon encounter them, lymphatic tissues are prominent in the connective tissue coats of the gastrointestinal tract, respiratory, and urogenital tracts. Lymphatic structures also are inserted into the lymph drainage and the blood circulation, where they serve to filter lymph and blood. This particular field illustrates the GALT, or the gut-associated lymphatic tissue, as you recall, that extends throughout the gastrointestinal tract, beginning at the esophagus and extending through the anal canal. This is a illustration of jejunum illustrating the GALT that lies between the intestinal glands and extends a little bit of a distance, perhaps, up into the lamina propria core of the villus. So this is a specialized type of reticular connective tissue filled with the reticular cells, fibers, and then superimposed on that background numerous lymphocytes, plasma cells, macrophage, and dendritic cells as well. So this is the GALT, the diffuse form of the GALT, or GALT, or gut-associated lymphatic tissue. This is a section of the ileum illustrating a portion of a Peyer's patch, which is part of this GALT system within the gastrointestinal tract. What this particular illustration shows is the development of an actual lymphatic nodule or follicle, with the germinal center being here, this lighter staining area, and then the close packedness density of the lymphocytes in the surrounding area. Then we run into more diffuse type of lymphatic tissue here, and then with another nodule, though poorly illustrated, occupying this area here, and just a nick through perhaps a germinal center appearing here. And then it continues on as the diffuse lymphatic tissue. So here we have a couple of aggregates or nodules constituting that entity known as Peyer's patch. Please recall that these organized lymphatic nodules can occur anywhere along the digestive tract, beginning in the esophagus. Please recall that there are scattered nodules there. Some would be found in the stomach. Some would be found at the gastrointestinal junction, throughout the intestine, down as far as the anal canal. So this is all part of this GALT system. This again is a segment of the intestinal tract showing the GALT, but in this particular case a segment through the appendix, and perhaps better illustrates these confluent lymphatic nodules. A nodule showing here with the germinal center now at the tip of the arrow. One can make out the diffuse aspect of the GALT scattered around between the crypts of Lieberkühn, or the intestinal glands. Here we get a section through a portion of a lymphatic nodule, as indicated by the arrow. Another one coming in, again indicated by the arrow. And here is a relatively large lymphatic nodule with a very robust germinal center as indicated by the arrow, and the nodule is now being traced by the arrow. This particular line here is just a fold in the tissue. The diffuse type of lymphatic tissue continues to be found between the intestinal glands. Now, please recall that you have under almost all the mucous membranes some sort of a lymphoid tissue, diffuse lymphoid system in place as a defensive mechanism. Generally, underlying all these mucous membranes, that is the gastrointestinal tract, urogenital system, respiratory system, these are all referred to generally as the MALT, or the mucosal associated lymphatic tissues. In the gastrointestinal tract, they're specifically referred to as the GALT, the gut-associated lymphatic tissue. And one last abbreviation, the BALT, as you will recall, in the respiratory system, in the trachea and the major bronchi, going down about as far as the cartilages extend, you'll recall, you can have lymphatic nodules in this region as well, and this is why it is referred to as the BALT, or the bronchus-associated lymphatic tissue. Now, lymphatic organs consist of accumulations of both diffuse and nodular lymphatic tissues that are organized into discrete structures. The lymphatic tissues that compose the organs are usually isolated from surrounding tissues by a well-defined capsule. Now, this particular field is from the palatine tonsil, so this is one of the tonsils, but for purposes right at the moment, note one can visualize a large lymphatic nodule here with its huge germinal center. Likewise, there's a lymphatic nodule again indicated by the pointer, and its enlarged germinal center located here. Likewise, there's another lymphatic nodule at this particular location with a very active germinal center here, another one associated here, and these lymphatic nodules are more or less set in a bed of diffuse lymphatic tissue. So as one peruses and courses around this particular structure, I think one can assume that we're dealing with some type of a lymphoid organ. So the question then arises, how does one differentiate between the lymphatic organs, that is, between various tonsils, the spleen, lymph node, and perhaps even the thymus? Now, it should be remembered simply that tonsils are aggregates of lymphatic nodules associated with the pharynx. They form a ring of lymphatic tissues in varying degrees of completeness that is present at the entrance of the esophagus and the respiratory tract, really, and are found in the oropharynx and nasopharynx, and perhaps even at the base of the tongue as the lingual tonsil. Therefore, we have a palatine tonsil bordering the oropharynx, a pharyngeal tonsil or adenoids sort of surrounding the opening to the nasopharynx, and then the lingual tonsils at the base of the tongue. The important thing to remember is that these lymphoid aggregates, that is the tonsils, which are partially encapsulated, are lying next to the lumen, and therefore are going to be associated with a type of epithelium, as indicated here. So, in this particular case, it's a stratified squamous type of epithelium. This is the epithelium as seen at increased magnification, a large patch of relatively thick stratified squamous type of epithelium of the non-keratinized or the wet variety. That particular feature, plus the tremendous development of lymphatic nodules associated with this region tells you that you're dealing with a palatine tonsil. This is the palatine tonsil seen once again with the scanning objective just coursing around it. Those two features, a confluence of lymphatic nodules associated with an epithelium, in this particular case a wet non-keratinized stratified squamous type of epithelium, tells you almost immediately that you're dealing with the palatine tonsil. We're just going to course around the tissue, keeping the epithelium in the corner and looking at the development of the lymphatic nodules, which is rather tremendous. Now, occasionally you run into structures such as indicated here that are filled with detritus and a bunch of debris. These are actually infoldings from the surface epithelium called crypts that extend all the way down to the base of the capsule. And we will see that the lymphatic nodules line up along these crypts, and they're actually surfaced by a continuation of the epithelium down into and lining the crypts down into the tonsil. In this particular case, the epithelium is so infiltrated by lymphocytes that it's pretty difficult to distinguish. So let's continue to course along the external surface. Here we can actually see one of the crypts entering in from the surface, and I'm outlining it and indicating it by the arrow. And you can see that it goes down a considerable distance into the tonsillar structure itself. As a matter of fact, we're down near on its basal surface looking at the capsule of the tonsil. Remember, the tonsil is only partially encapsulated, only along its basal lateral surfaces, and that the luminal surface is going to be covered by not a capsule, not a connective tissue capsule, but by the covering epithelium wherever it is. And here we can see some compound tubular alveolar gland, just some pharyngeal glands just beyond the connective tissue capsule of this particular structure. Then we have some more lymphoid tissue there. So this is a lengthwise cut through one of these crypts. If one looks right here at the edge of the arrow, one can make out a stratified squamous non-keratinized type form of epithelium. All of these features indicate that this lymphoid structure or organ that we're dealing with here is the palatine tonsil. So aggregates of lymphoid tissue, partially encapsulated, exhibits crypts lined by a non-keratinized stratified squamous type of epithelium, tells you it has to be the palatine tonsil. Once again, this field represents a concentration of lymphatic nodules, one shown here, with extraordinarily active or hot germinal centers, and then the little cuff of lymphocytes. And once again, note that it is lined or covered by or surfaced by an epithelial type. This tells you immediately that you're dealing with a tonsil of some variety. However, as we course along, look at this particular epithelium in particular. It is not a stratified squamous type of epithelium, but indeed looks like a pseudostratified columnar type of epithelium, indicating that this tissue was taken from the nasopharynx. Therefore, it should be an adenoid or a pharyngeal tonsil. The epithelium seen at an increase in magnification strongly suggests that yes, indeed, this is a pseudostratified columnar type of epithelium, and if one went down at increased magnification, cilia would even be observed. This is an interesting case because if one peruses or moves along a little bit further where there's more of a lymphocytic infiltration of the epithelium, note that the epithelial surface has changed to a stratified squamous type, and indeed as we go over to this particular region, it is indeed lined in part by a stratified squamous type of epithelium. This is not unusual, particularly in people with chronic allergies, postnasal drip, aging, chronic smokers. You get a metaplasia of the epithelium going from a pseudostratified columnar epithelium, as is shown here. It'll become metaplastic and transform into a stratified squamous type of epithelium. So once an epithelial surface is located, the entire surface should be looked at, and if one finds groups or islands of ciliated pseudostratified columnar type of epithelium, one can indeed assume that it's from a pharyngeal tonsil or adenoids. Here we can see once again it's metaplastic and has changed in its epithelial lining. So one has to be a little bit cautious when examining the epithelium, course through and examine the entire surface, and then look at the epithelium lying in a crypt. Oftentimes the epithelial surface within the crypt is somewhat protected from those events on the surface, and that will give a good indication of what the true epithelium associated with the lymphatic aggregate is, and then one can determine specifically what tonsil one is dealing with. This is a section of lymph node as seen with the scanning objective. One notices immediately its characteristic features: an outer limiting capsule of collagen, though thinner than might be anticipated, an underlying subcapsular sinus. One can make out in this particular area a relatively thin cortex. Here is a fairly good sized lymphatic nodule with a germinal center, and if one courses to the interior portion of this region of this lymph node, one can make out medullary cords, which will be filled with lymphocytes and plasma cells, perhaps a few macrophages, and then surrounding the medullary cords are, of course, the medullary sinuses. If one courses even further, one can continues to notice the outer lying capsule. These little projections coming in here and about this point are trabeculae that are going to compartmentalize and forming a sort of a skeleton or support for the reticular type of connective tissue that will fill in the background, and the lymphocytes will eventually be superimposed upon. Here again we have the capsule, a trabeculum coming in here, and then the subcapsular sinus in this particular case appears to be filled with lymphocytes coursing along that region. This vessel here is probably an afferent lymphatic vessel, a portion of a little valve here that will eventually drain into the subcapsular sinus. And once again, a trabeculum entering and then going out of the field of view, as is the case at the tip of the arrow. Here's a little bit better presentation of what one hoped to see. The outer thin capsular area here, subcapsular sinus, crisscrossed with reticular processes and fibers. This would be a trabeculum entering through the cortex and then extending down, subdividing and forming the base skeleton that will help support the reticular tissue of this particular lymph node. And one can also see in this particular section these flat sinuses are continuous one with the other. So this is a subcapsular sinus. This lying on either side of the trabeculum are the trabecular sinuses. And then finally one can see a portion of the medullary medulla, these being the medullary cords, medullary sinuses running on either side of the medullary cords. The medullary cords being consisting of a diffuse type of lymphatic tissue, as one will recall, filled with plasma cells and small lymphocytes. Now we're coursing more towards the hilar area of the lymph node, which is the entrance point of small arteries or arteriole size. This is probably the hilar region here, and the exit point for the lymphatics. Afferent lymphatics come around the sort of the circumference of the lymph node. And then this hilar area, after the lymph has percolated through the subcapsular sinus system, the trabecular sinus system, and then in this medullary sinus system, will eventually collect and leave the lymph node via one or two efferent lymphatic vessels, one of which is shown here at the arrow. And then this will repeat itself again and again. So once again we have or in this particular case, for this structure, we have a capsule with trabeculae extending from the inner surface of the capsule, and then linking to it. These trabeculae extend down even though they go out of the field of view, and partially subdivide but don't completely compartmentalize the interior of the lymph node and support the framework where the reticular fibers and cells attach to. Having established that framework, then the lymphatic tissue is placed upon it. The nodular lymphatic tissue, where the B cells will hone in on, forms this outer type of cortex. There is also a deeper cortex where the T lymphocytes seem to reside, but this lymph node doesn't seem to be reactive to demonstrate that very well. It would be a sort of a diffuse non-nodular form in this area if it were present. And then you also have the diffuse lymphatic tissue forming the medullary cords. And then that particular region consisting primarily, as I said before, plasma cells and small lymphocytes. This is a portion of an additional lymph node, one which has been reacted with the silver stain, but nonetheless shows several important features. The capsule is located here at the tip of the arrow. Again, the trabeculae extend from the inner surface of the capsule down into the substance of the lymph node. One can see just beneath the capsule several lymphatic nodules more or less lined up like they should be. This would be the nodule here in this particular case, and the germinal center staying darker in this particular instance. And the outer cortex goes to about this level with the nodular type of lymphatic tissue. With close observation, one can make out just barely a subcapsular sinus, trabecular sinus to repeat once again, and then the medullary sinuses in this particular area, and the medullary cords just barely starting to form or come in. This region here would be the deep cortex, just prior to being this diffuse type of lymphatic tissue, just prior to its forming into medullary cords. So this is the thymic dependent area right about in here of most general lymph nodes, if it were put like that. This area will almost disappear following the thymectomy, at least in experimental animals. This is a reticular stain, as I mentioned before, to demonstrate reticular fibers that are going to form the supporting framework of the free cells or the lymphocytes themselves. And it can be demonstrated quite readily if we examine this particular preparation at increased magnification. This is a region of diffuse lymphatic tissue as seen at increased magnification and treated with the reticular stain, or the silver stain, showing the reticular fibers which stain black and form the stroma of this particular organ, with the parenchyma being the small and medium-sized lymphocytes that form the parenchyma of this particular structure. So one gets a much better idea by using some of the special preparations to demonstrate some of the features, these features of the lymph node. This is a region through a medullary sinus, showing just at the very edge just a portion of a medullary cord, now indicated by the arrow. But what one is trying to demonstrate here is the sinus system as seen at increased magnification. Now it makes little difference whether you're in the subcapsular sinus, along the trabecular sinus, or in the medullary sinus. They're all pretty much constructed the same way. This particular field shows some of the reticular cells quite well, one shown here, one here, one here. Remember, these particular cells are nothing more than modified fibroblasts that are specifically modified or function to produce and maintain reticular fibers. And so across this sinus space, in this case a medullary sinus which is coursing, let's say, in this direction, one will see that it's crisscrossed with fine processes of these reticular cells, and within are sort of forming a round. The cytoplasm is actually around a fine reticular fiber that's bridging this gap, or these sinusoids. So as the lymph flows through them, they form these little eddies. They have to go over and around, and it breaks up the flow to slow down the flow through the lymph nodes. And it's really not a flow like one would see in a channel such as one of the vessels, but it more or less oozes through these lymphatic sinuses, so it can be more carefully and thoroughly monitored. Now, as one will recall, numerous macrophages are found within this medullary sinus system, and will actually phagocytize and take up particulate matter. Obviously, there are numerous small lymphocytes that are also flowing through, though most are housed within the cords, the medullary cords, which are absolutely filled, as one will recall, with plasma cells and small lymphocytes. If we just go up to and look for review purposes in the structure of this medullary cord, one can see the numerous plasma cells. You recall this is one of the places that they were examined in detail, as well as the few scattered lymphocytes and macrophages as well. The main feature was to demonstrate how broken up this channel is, the sinus system, and it's crisscrossed by these reticular fibers and overlying reticular processes of the reticular cells producing and maintaining them, and how this flow of lymph through these channels sort of percolates through, and how closely it can be screened and monitored, both for particulate matter and for antigens in this particular region. Now, it makes no little difference whether we're looking at the subcapsular sinus, as I mentioned before, the trabecular sinus or the medullary sinus, they all have this same structure that breaks up this flow, and there's it need not be demonstrated any further in this particular exercise. This once again is the hilar region of a lymph node, or at least pretty close to it, seen with the scanning objectives. Two efferent lymphatic vessels here, one which appears to show a bit of a valve there. Unfortunately, there are no blood vessels in this particular area. We just have a lot of disrupted blood. The vessels were probably torn when this lymph node was gathered. Nonetheless, as we go into the medulla, realizing that even though this is an organ that actually filters the lymph fluid, it's large enough and needs its own vascular supply. The arteries, small arterial size, will enter at the hilar area, enter into the substance of the lymph node, and likewise small veins will drain and exit through this hilar region. That's the usual pattern of flow. Now, as one enters into the substance of the lymph node itself, and if we stop and at this particular field here, one can see that the small arteries, about the size of an arteriole or maybe a smaller caliber artery, make their way into the substance of the lymph node by coursing within the trabeculae, which are shown scattered out and form this inner supporting framework. They come from both, as you'll recall, the inner aspect of the capsule and extend up from the medulla or the hilar area as well. Here's an exiting small vein, again within this trabecular system. The vessels then will run up, as one can see here, through this system, leave the trabeculae as a series of capillaries, form loops, and then come back down the capillaries, forming then venules, and these venules will then coalesce, combine with one another, and then enter trabeculae, and the process reverses itself down to the hilar area. Now, in the region of the deep cortex, which would be right along in this region, the venules assume a rather unusual profile, and I don't know whether we'll be able to demonstrate in this particular lymph node or not, in that the lining endothelium in the deep cortex becomes rather exaggerated to form either a cuboidal or a columnar type of cell lining, and these are called postcapillary venules and are characterized by this very tall lining endothelium. This is a portion of that deep or paracortex seen at increased magnification, or the highest objective that I have, the very highest power, to show some of the postcapillary venules. A fairly decent one is shown here at the arrow, made up of 4 endothelial cells. You can see that they pretty much occlude the lumen. It looks like a lymphocyte here is just having egressed and passed through this type of endothelium. There are other examples shown here. Here's a fairly disrupted one. Another is shown here, and another is shown here. So this is the appearance of these postcapillary venules in the deep cortex of the lymph node. This is the region near the capsule of a lymph node taken with the intermediate objective, simply to illustrate the capsule as indicated by the arrow, the subcapsular space or sinus now indicated by the arrow, and then a very prominent lymphatic nodule that illustrates quite well its organization. The nodule extends out about this far. I'm trying to trace it with the pointer, set in a little bit of diffuse connective tissue, but what is so well circumscribed is a very active germinal center, which extends about this far. You see it's very, very well circumscribed in this particular case. So it's a very, very active lymph node. And then as we go down into this region here, this would be the deep or paracortex, and then finally moving into the medullary region here with the medullary cords indicated here, and then the medullary sinuses as indicated by the arrow. So that is a pretty well developed and active lymphatic nodule, and the germinal center of which almost fills the field of view. This is a second of another of the lymphatic organs, the spleen. Please recall that the spleen, unlike the lymph node, does not consist of a cortex and medulla, but is divided or segregated into two major subcomponents, the white pulp and the red pulp. The white pulp refers to the accumulations of lymphatic tissue and stains of basophilic color because of the huge concentration of lymphocyte nuclei associated with it. And then the red pulp, which is a diffuse type of lymphatic tissue rather than a more concentrated nodular form, and it is, because it's associated with the venous component, absolutely filled or suffused with red blood cells, the erythrocytes, giving it the reddish hue or color. So that is a major distinction. Like the lymph node, the spleen is surrounded by a rather robust capsule made primarily of collagenous or collagen type of connective tissue with fibroblasts. Occasionally a few smooth muscle cells are found in it as well. But let's just course along and look at the similarities and difference, contrasting and comparing lymph node structure with that of the spleen. The capsule, once again, is located where the tip of the arrow is. Like the lymph node, the spleen has rather large trabeculae, these big bars of connective tissue. One is just coming into the field here, just a little cut portion of it, then it goes out of the field, and they will extend to the interior of the spleen. And like the lymph node, there is a reticular type of connective tissue made up of reticular fibers and reticular cells, those modified fibroblasts that produce the reticular cells that produce the supporting framework, the stroma, of which the lymphatic tissue is embedded on or held upon. This region here would be described as red pulp, white pulp showing here. If we course along, here once again we see just a portion of the capsule, a trabeculum, a portion of one meandering across in this direction. This would be the white pulp, another portion of the white pulp, the background material being considered red pulp. Another little portion of a trabeculum, we're just nicking it, coming from the capsule, extending inward to the interior of the capsule. And remember, strung across between trabeculae is this reticular fiber cell network that is going to form the stroma of this particular organ. a cross-section of a trabeculum here, a section through another trabeculum at this particular location. Once again, capsule, trabeculum, it's splitting here, red pulp, white pulp, white pulp. And here we get a bit of the capsule and showing a trabeculum extending a little bit further along. Another section of spleen, this time more towards the interior of the spleen, once again viewed with the scanning objective. Now, it is very helpful to realize that the interior of the spleen and its segregation into the red pulp and the white pulp is intimately related to the different parts of the vasculature. The white pulp is related to the arterial portion of the spleen, whereas the red pulp is directly related to the venous side and the sinusoids, the splenic sinusoids of the spleen. Now, the arteries, if we trace beginning with the arteries into the interior of the spleen, enter from the capsule, from the hilar area, and run in the trabeculae. Here we can see a portion of a trabeculum and two small arteries approaching the size of a small arteriole within this trabecular framework. So this would be a trabeculum as shown here, with a trabecular artery within its interior. Red pulp located here is indicated by the amount of red blood cells within it. A portion of white pulp, white pulp, white pulp, and this is more of the red pulp area. Now, if one traces the trabeculum and the contained trabecular artery, here we can see the trabeculum coursing this way, and even with the scanning objective, one can make out the artery wall, the smooth muscle wraps here. So this is indeed a trabecular artery. We'll examine a trabecular vein momentarily. And eventually, what will happen is an artery, or a branch of the trabecular artery, will leave the trabeculum, as is indicated here, and course into the pulp of the spleen. As it does so, it immediately picks up a sleeve of lymphocytes, rather intense in their concentration as showing here, and this is the periarterial lymphatic sheath, or the PALS. So it's this arteriole, this central artery, but actually, in actual fact, classified as an arteriole, that the white pulp is intimately related to. Initially as the periarterial lymphatic sheath, as is shown here, and then somewhere along its length, oftentimes a clone of B lymphocytes will develop and a lymphatic nodule, or a Malpighian corpuscle, will develop. So you do have splenic nodules, lymphoid follicles associated with this periarterial lymphatic sheath, and also forming a portion of the white pulp. Now, this former trabecular artery, this would be a central artery here, picking up its pals and curving around in this direction. Then it'll become a smaller vessel that will subdivide into small penicillar arteries. Penicillar referring to the tip of a paintbrush, so you get a few strands that reminded the earlier investigators of a paintbrush. They too will have a little bit of a sleeve of lymphocytes, maybe 2 or 3 cells in width, and those will be the penicillar arteries, which we'll examine momentarily at increased magnification. Nonetheless, the central artery came in this direction. If we trace it a little bit farther, one can see the pals associated with it. Here we're probably just getting into a portion of a lymphatic nodule because of this extension at this particular location. And eventually the penicillar arteries will form a series of capillaries as it ends, and oftentimes, not all of them, but many will pick up a sheath of reticular fibers and phagocytic type cells, and these are called the sheath capillaries. Those are the primary elements one can visualize, though many others are described in these sections of spleen. So if we just look at this particular field here with the scanner, one should be able to discern the following. A section of a trabeculum here, this region here being the red pulp due to its reddish type color. The white pulp is the more dense lymphoid tissue, as one can see here. This is all white pulp. In life or in autopsy, if it would have been cut, because these are leukocytes, the white cells, a family of them, it would have that whitish hue to it, like little pustules, hence its name from autopsy, the white pulp. The red pulp, of course, would have been stained a deep burgundy, reddish type of color. And in this case, with the stains, the red cells take on that red color. So that's what can be seen in this particular field. And once again, we can see, though we don't see the continuity of the vessel, white pulp, these would be the central arteries here. By no means central, always have an eccentric position, or almost always, in the periarterial lymphatic sheaths. And centrally, if you're approaching a nodule, which this appears to be happening here, more often than not, the central artery will have a eccentric position within the nodule or the white pulp. It simply left the trabeculum and entered the center of the substance of the spleen, hence her name central artery. So this would be a trabeculum with a trabecular artery, a branch of the trabeculum coming here. This would be the central artery in several sections, and the pals, this lymphoid sheath associated with it of dense lymphoid tissue, and perhaps even just beginning into a lymphatic nodule at this particular location. So now let us go to an increase in magnification and look at the vessels that can be seen usually in a section of a spleen at a slight increase in magnification. This again is the spleen at a slight increase in magnification, showing the trabeculum occupying the center of the field, a branch going off in this direction. To illustrate, if one looks at it more carefully, one can actually see the muscle wall, the tunica media, of the trabecular artery, which is cut in cross-section here. Very, very close observation. You can even make out the internal elastic lamina at this point. Then the muscle wall is cut a little bit tangentially, and the vessel is coursing in this particular direction, the direction of the arrow. At this particular juncture, this would be red pulp at this location, red pulp at this particular location. Now if we continue to peruse along, and all of a sudden we can see that this vessel begins to split. This will be becoming a central artery in this direction as it leaves the trabeculum. Trabecular artery going in this direction, even though it's an oblique cut. And one can see as it does so, it immediately picks up a relatively robust sleeve of intensely packed lymphocytes. So this is the pals of the periarterial lymphatic sheath, that portion of the white pulp coming in, and that's what we're tracing along here. So this is white pulp, and specifically the periarterial lymphatic sheath. And if we course along this particular vessel, we can see it coursing in this direction. Again, the pals located here and here. We may be approaching a nodule here. It's getting very dense. This would be red pulp on this side from about where the pointer now indicates, and to the left, this is all red pulp, easily discern, due to the red color, the intense concentration of lymphocytes. And then finally, we're out of the field, but the central artery or branch thereof coursed in this direction, but it's out of the field. Then all of a sudden, a much smaller caliber arteriole comes into the field, and then we'll loop down and course in this direction, though we're only seeing one of them at this particular junction. At about this location, when it becomes small, much smaller in diameter, it picks up a sleeve of just 2 or 3 layers of lymphocytes. This is one of the branches that'll be very close in appearance to one of the penicillar arteries. This once again is the spleen, as we're looking at the vascular, seen at a slight increase in magnification, with the para-arterial lymphatic sheath encompassing this area. So this is an area of white pulp, red pulp around in this area. And what this is illustrating is the central artery coming out of the pals, probably very close to a lymphatic nodule as this appears, but then subdividing here, and this is roughly the point where we get a splitting into the penicillar arteries, these small vessels. One which is coursing here, as we're seeing at a slight magnification, and one can see that that is at the location of an arteriole, or the size of a arteriole, excuse me. Another branch appears to be going over in this direction. So this is how they appear as the arterial portion of the spleen ends. And with its ending, so ends this intense concentration of lymphoid tissue that's organized along its pathway, the white pulp. Another region to the interior of the spleen, showing a central artery here, the para-arterial lymphatic sheath here, forming a portion of the white pulp, and then finally red pulp to the left of the field. Now if we course up in this particular direction and into this faint area of white pulp, just where it's disappearing, one can see several branches of this arterial system. One shown here, one shown here. I've got the pointer in the lumen. One shown here, another shown here. And see, there's 2 or 3 layers of lymphoid cells associated with each one of these branches. Now, these are all penicillar arteries, these 4 branches. And one can appreciate what the early investigators were talking about, what they related things to. If we go back to this central artery here, it perhaps coursed up out of the field to this direction, came up, and then about this location, somewhere behind the arrow, split into its 4 or 5, maybe 6 branches into these terminal arterioles, continued to take their little sleeve of lymphocytes with them, and then will become capillaries, some of which will be enveloped by the sheath capillaries of the spleen, which will then end in the red pulp. Another region of the spleen seen at increased magnification, where we have the sort of the end part, a lengthwise cut through a central artery. So this is white pulp here, a little bit of white pulp, red pulp in this particular area. But notice this vessel. The central artery is ending. We're getting a sort of a lengthwise cut, and this is perhaps will help your understanding a little bit. This vessel will then very quickly branch into 4 or 5 little branches. So these would be the penicillar arteries, still containing a little sleeve of 2 or 3 lymphocytes around them, and then we'll finally end in sheath arteries. And we'll take a little, this could be one down here, we'll take a little bit of time to look for one. So this is one vessel going here, one going in this way, and perhaps another one heading this way before they end in these these sheath capillaries, or just plain capillaries within the red pulp. This particular field is taken at high magnification from the red pulp of the spleen, and what's being illustrated here is after the penicillar arteries lose their sheath, they too branch into a series of short capillaries, one of which is cut lengthwise along its axis, as shown here, coursing in this direction. These are the endothelial cell nuclei lining the lumen, and some of them are cut a little bit tangentially as we got through the vessel. Let me attempt to focus this in and out to get a better appreciation of this structure. So this is one of the capillaries as the arterial system of the spleen ends. Now, unfortunately, this one doesn't show a wrap of the sheath. These phagocytic cells and some of the reticular cells form an encapsulation or a sheath area around the external wall of these capillaries, hence they're called sheathed capillaries. We may or may not be able to find one of these to be demonstrated, but nonetheless one can see the caliber of how this portion of the vascular system ends, that is the arterial system. Now this is all red pulp on this side, red pulp on this side. The red cells are coursing within this diffuse type of connective tissue, and now the next problem that one will face is to find the beginnings of the venous side, that is the venous sinusoids of the spleen. Again, the interior of the spleen, looking at the red pulp, this is a poorly demonstrated one of the sheath capillaries. You can see the minute lumen and then just a little bit of a reticular sheath around it. This gives you an idea, however, what these particular structures look like. Now, the last thing one should look for before leaving the examination of the white pulp is look for the marginal zone. A small zone, very obscure. One looks for elongated fibers and nuclei of fibroblasts, is when I'm trying to point these out, that forms the boundary, or the margin as its name indicates, that sort of demarcates or separates the white pulp, which is shown to the left, and the red pulp, which is shown to the right. One will see a very fine organization along that perimeter. And this is how it appears. Extraordinarily vague, but nonetheless it is there. And this is what one looks for, is these elongated nuclei and a few strands that separate these two arenas of the spleen. This is that same marginal zone seen at high magnification. This is the region we're trying to indicate to you, is these elongated nuclei and little fibrous strands that form this boundary. It extends from here, where the arrow now indicates to this location, sort of a interface between red and white pulp. There's a definite organization along this boundary between the two pulps, but it's very obscure and difficult to visualize in section. But nonetheless, this is what one is talking about when they're referring to this marginal zone between the red and white pulp. Now, having made a careful study of the arterial portion of the spleen, that portion of the spleen intimately associated with the white pulp, which is shown here, and remembering that the dense or the nodular lymphatic tissue is associated with the arterial portion of the spleen. Having finished that exercise, now examine the red pulp, that portion of the spleen that is associated with the venous system and venous return from the spleen, and it begins as a series of sinusoids within the red pulp. Perhaps some of these lumina are these sinusoids. It is a difficult exercise, particularly if the spleen is suffused with erythrocytes, which it usually is. But with a bit of persistence, one can usually find an area that can indicate where these sinusoids are, and one can trace the migration of the erythrocytes and leukocytes through this diffuse lymphatic tissue that forms the background or the splenic cords within the spleen, and watch these cells enter into the lumina of the venous sinusoids. So that is the next exercise, and we'll look for an area that hopefully will illustrate these features. This is a section of the red pulp, but of a plastic embedded specimen. I chose to use this one for demonstration purposes because it so much more crisply illustrates the sinusoids in the red pulp of the spleen. Remember that these sinusoids consist of endothelial cells, elongated endothelial cells, and they're arranged much like staves in a barrel or segments of an orange, though the cells are not in direct contact necessarily with one another, and they're loosely held in three-dimensional space by bars of basement membrane. Not sheets, but bars. So there's a loose wrap of discontinuous basement membrane around them, making up or holding them in three-dimensional space. Now, these are the bars of basement membrane right at the tip of the arrow. I'm looking at this specimen under the highest magnification that I have. So this is the outside of the sinusoid. The lumen of the sinusoid is shown here. So all of these endothelial cells are cut in cross-section, and we're looking at them in a transverse profile. So this is a nucleus of an endothelial cell. One is shown here, one is shown here. These red structures, of course, are erythrocytes, and one can see outside of the sinusoid there are some lying here, one coursing here, and this one, it looks like it's squeezing between and it's going to enter the lumen. We can kind of see this if we look at this sort of elongated sinusoid. I'm tracing the base of it. It goes around, wraps around, and then goes up sort of like that. If you look carefully along here we have some more erythrocytes in that diffuse lymphatic tissue, squeezing its way through, squeezing between the endothelial cells, and then finally entering the sinusoidal lumen here. So each one of these structures, and one can make out just a little wisp of the indicated by these bars of basement membrane, making the perimeter or the outline of these sinusoids a little bit more easily to visualize. Here we can even make out a couple of beads, these little bars of basement membranes, how they really appear. This would be another venous sinusoid here, and the red cells continue to move and press between these endothelial cells to gain access to the venous return of the spleen. So this is actually a very good area, or very good preparation to illustrate that. This is difficult to work with. On the preparations within the slide collection, the paraffin section, one has to rely on a collection of endothelial cell nuclei. So what one looks for is several nuclei in a pattern, and one can vaguely make out the external surface and separate the lumen from red cells and leukocytes in the surrounding reticular type of connective tissue. Be sure one compares these, and I will demonstrate one very shortly, how these sinusoids appear in a scanning electron micrograph, because this will give you a more precise three-dimensional image of these particular structures, which are really difficult to visualize in some of the two-dimensional views you see in the thin section. This is an elongated profile through one. If I trace its basement membrane through a sinusoid, it comes down and then it will come back like so. You can see this one has filled with some leukocytes, and red cells making their way through to gain access to the lumen once again. So this is a plastic embedded material through the red pulp of the spleen, illustrating its venous sinusoids. This is an artist's depiction of one of the venous sinuses or sinusoids from the spleen. And what this particular line drawing illustrates quite well are the stave-like endothelial cells. So these endothelial cells are organized more or less lengthwise to one another, separated by spaces, and are united in three-dimensional space by these bars of basement membrane, as indicated by the arrow. Most often when one views these in sections, that is the sinusoids, one gets a cut as shown here. And what one looks for, if the section were taken at this angle, is sort of a ring of nuclei with a bit of cytoplasm around the nuclei, and this space would be filled with red cells. And so one looks for a line of those nuclear profiles to determine the sinusoidal wall. It is indeed difficult when initially looking at the red pulp of the spleen. This gives a very good illustration of what the venous sinuses in the red pulp of the spleen are visualized to look like. This is a scanning electron micrograph through the red pulp of the spleen, indicating or demonstrating the interior surface of the venous sinuses or sinusoids. Here is one located at the arrow. The lumen, of course, is here. These would be the endothelial cells arranged in this stave-like pattern. So this is the lumen of a sinusoid. Another sinusoidal lumen is shown here. It's coming out of the field, it courses and then goes down in this direction. Another branch, continuing branch, goes up in this direction. This material here are all lymphocytes, red cells, and a reticular connective tissue. So this is the substance of the red pulp, that diffuse reticular type of tissue, and located here as well. And so the red cells, such as here, would be coming from the outside and then squeezing through this sinusoidal wall to enter and collect into the lumen of the venous sinus of the spleen. So this gives a very good example of the endothelial cells, how they're arranged in these particular venous sinuses in the red pulp of the spleen. A portion of a wall of one of the venous sinuses in the red pulp of the spleen as seen at increased magnification. So these are the endothelial cell bodies here, the cytoplasm, as one can see. Two red cells, and this would be the fractured surface. This is the diffuse lymphatic tissue here. One can make out a red cell here that'll probably course through, insinuate itself between the two endothelial cells, and then enter the lumen. Cells such as these are lymphoid cells. There are also numerous macrophages, of course, in this surrounding diffuse lymphatic tissue to take out the worn out or damaged red cells and leukocytes as well, as this is where they're going to be screened out and gotten rid of at the end of their lifespan. The venous sinuses or sinusoids will then be empty into or anastomose with and collect into veins of the pulp, a longitudinal section which is shown here. They're lined by more or less a continuous type of endothelium and may be supported by just a few smooth muscle cells that form sort of an obscure open spiral around the supporting the endothelial cells. So they're indeed a very thin-walled vessel as well. This is an example of one of these veins of the pulp, as indicated by the arrow. These in turn will then become continuous with and join with the trabeculae and will form the trabecular veins of the spleen. Another vein of the pulp, the lumen of which is showing here. Here we're just nicking its wall, but it's nice and open and clean. Here you can see a few endothelial cells and a supporting smooth muscle cell. So this is gives one a fairly decent idea of what the veins of the pulp appear as or look like. Here we can see one of the wraps of smooth muscle coming in and helping it, making its identification easier. And one can also see the endothelial lining, the continuous endothelial lining of this particular space. So this is a fairly decent example of one of the pulp veins. Remember, these are going to anastomose together, enter a trabeculum, and then will be referred to as the trabecular veins. This is another view of the spleen with the intermediate objective illustrating a trabecular vein. This is the trabecular wall here, and we can see it go out of the field. Note that in this particular case there's no smooth muscle wrap. It's more or less a fine endothelial line space, perhaps with a little bit of a supporting wall of the venous system. But by and large, most of the wall is made or consists of the trabeculum itself. Return and compare trabecular artery with a trabecular vein. And in the artery, one will definitely see the smooth muscle wraps associated with the trabecular artery. In this case, these cells or nuclei thereof are simply fibroblasts within this dense band of collagen forming the trabeculum itself. The vein is more or less an endothelial line space coursing within these particular structures. This is another of the lymphatic organs, a thymus, a section of a thymus, a fairly young thymus, perhaps from a juvenile individual. Note that it is readily distinguished or can be distinguished by an outer lying dark cortex, dark staining, and a centrally positioned lighter staining medulla. So in that regard, a cortex and a medulla, it is similar to the lymph node, and unlike the spleen, which was characterized by red and white pulp. The second feature to notice that this is the only lobulated lymphatic organ or structure where lobules can be discerned on the outside. Those perhaps weren't the best. Here would be more of a complete lobule with its cortex and a central extending medulla into the center of that lobule. And here you can once again appreciate that sort of lobular nature to this particular lymphatic organ. Now if we go back to the region we initially started with, at this region, the entire thymus is enveloped by a very thin capsule, and it does have little septae that extend down into the substance of the thymus itself. Like the spleen and lymph node, there is a reticular network on which the free cells, that is the lymphocytes, impinge. But in this particular case, it's somewhat different. These are of endodermal origin, that is the reticular cells, and what we're dealing here is something totally different than from the lymph node and the spleen. The thymus, the supporting stroma or framework of the thymus, is a cytoreticulum. made up of these reticular epithelial cells derived from endoderm. It is not supported by a reticular cell and fibroblast type of cell of mesenchymal origin and reticular fibers. This is a cytoreticulum and is unique to the thymus. What we will now do is examine some of the details of the cortex, then proceed to examine some of the finer details of the medulla. Before proceeding, however, note that the medulla is continuous up into these lobules, and the thymus is easily distinguished from the lymph node, which also has a cortex and a medulla, but there are no lymphatic nodules with germinal centers. A lymphatic nodule and germinal centers indicate the B lymphocyte cell line, its type of activity, or is associated with those nodules. One would not anticipate those being where the T lymphocytes are made, that is, namely in the thymus, where they're taking their origin from, and this is indeed the case. There are no lymphatic nodules and germinal centers within the thymus because the lack of B lymphocytes. In those areas, and let me see if I can find one, that do give that general appearance, such as this structure here, if one considered this a nodule and this lighter area, germinal center, are a little bit misleading. This is simply an extension up of the medulla into the cortex. The medulla is continuous throughout its extent and has projections that extend up into these lobules, sometimes giving the impression of a isolated lymphatic nodule. But in actual fact, if the section could be viewed in its entirety through serial sections, one would see the medulla is actually extending up. And these are lobules such as here that appear as isolated entities, but this medulla would eventually, either coming out of the section or going into the section, come back up and be continuous with this. So this is a little bit misleading. But one must remember this: the function of the thymus is not the filtration of a lymph fluid. It is not the filtration of blood. Its primary function is the production of lymphocytes, namely the T lymphocytes. And so one would not expect that nodular or lymphatic nodules with a germinal center, even though it does have a cortex and a medulla. So now let us examine the cortex initially and see what microscopic features can be observed in that particular region. This is the external surface of the thymus, or the cortex of the thymus, seen at high magnification, indicating the capsule in this particular region. The outer regions of the cortical area of the thymus show a lot of blast-like cells or medium-sized lymphocytes. I don't see a lymphoblast at the moment, but one can get an idea of what large or medium-sized lymphocytes appear as. They have this lymphoid character, but they're much small or larger than the small lymphocyte. So this would be one of these medium-sized lymphocytes. Another one here, one here, one here. The nucleoplasm a little bit more euchromatic, and the cytoplasm more abundant, and they are quite a bit larger than the typical small lymphocyte, as indicated by these relatively dark nuclei. Some of us that examined this actually under the microscope earlier will recall that reticular cells can also be found in this particular region, as well as macrophages associated with some capillaries that will loop up in this area. And also, because a tremendous amount of mitotic activity is taking place in this region for the production of T lymphocytes, numerous mitotic figures will also will be observed in the background, though they're quite small and difficult at times to make out. One is shown here at the tip of the arrow. as we move deeper into the cortical area, more and more small lymphocytes are observed, with the medium-sized and the larger ones becoming less abundant. Numerous mitotic figures continue to be observed, as indicated here and here, over here. So a tremendous amount of activity has taken place. These large empty spaces oftentimes indicate the position of a macrophage, so the nuclear profile isn't exhibited very well in this one, just a shadow of it. This is probably a lymphocyte that's been phagocytized. Considerable amount of cell death takes place in this region, as well as mitotic activity, as those self-recognizing lymphocytes that are self-recognizing, recognized cells, are taken out and destroyed by macrophages in this area. Another mitotic figure, another one, another one, another one here. So one can get a fairly decent idea of the tremendous amount of mitotic activity for the production of these T cells. And here's perhaps the edge of a macrophage nucleus, and then the remnants of a broken down lymphocyte within its cytoplasm. Cytoplasmic boundaries, perhaps, in this macrophage extend about thus far. These large light staining cells, such as here, here, and here, with a rather crisp nucleolus coming in, are those cells that belong to the cytoreticulum. One is just seeing their nuclear profile, and their cytoplasm is in the form of very thin processes or sheets that, in this particular section, are almost invisible, as they are sandwiched between this close compacted network of small T lymphocytes. Now, as one leaves the cortical region of the thymus and enters the medullary region, obviously the section becomes lighter. Some of the reticular cells, such as shown here, become more obvious because they're much larger cells. The nuclear profile stands out. It doesn't have the lymphoid character. And oftentimes the reticular cells will start forming into whorls to form thymic corpuscles or Hassel's corpuscles. Here we're just getting into the edge of one of those particular cells or organizations, showing reticular cells forming into these whorls. This perhaps is a little bit better one. So this structure here, starting to form some concentric whorls, is some whirling of this Hassel's corpuscle, or this thymic corpuscle. The corpuscle extends from here to here, here to here. And they may be quite large, even becoming hyalinized or a little bit necrotic in their centers. Here's another portion through one of these Hassel's body. And here's one that's a little bit larger, a typical Hassel's corpuscle, and made up of this whirling up of the reticular cells. So if one studies the Hassel's corpuscles initially, looking at the nuclear profiles in particular and the light cytoplasm, their size, then using that profile or that information to go back up into the cortical or the medullary region, whatever region one wants to look for or examine reticular cells, this gives you an idea of the nuclear profile, which one to look for, because these Hassel corpuscles are made up of these whirlings of the reticular cells. That smaller whorl we initially looked at is located here. Here we can see some reticular cells that are forming part of the cytoreticulum. This again is the medulla of the thymus, seen at less magnification using the intermediate power, showing Hassel's corpuscles that are fairly characteristic of the thymus, or sometimes referred to as thymic corpuscles. Here are a couple of more in the medulla, and perhaps shows a little bit better the concentric whirling nature of these particular structures. Another one is shown here right at the tip of the arrow, and it's become hyalinized and maybe even a little necrotic in its center. So that gives you an idea of what these Hassel's corpuscles really look like. Now here are a couple more at this location, and another one shown here that looks like it too is degenerating somewhat. So these are the thymic corpuscles, or Hassel's corpuscles, oftentimes considered characteristic of the thymus. This is simply another Hassel's or thymic corpuscle, a very large one in the medulla of a thymus. So they are all different sizes, from quite small to some that are extraordinarily large. This is a paraffin section of adult human thymus. The capsule or limiting capsule is located in this particular area. So what we're looking at here is the cortex. Notice that it is not nearly as active in the production of T lymphocytes or small lymphocytes. And as one courses down towards the medulla, which one gets to very quickly, one can visualize once again several Hassel's corpuscles, or these thymic corpuscles, in the medullary area. So it's filled with these types of structures. And then again we're coursing a little bit more along the medulla, and then go back out towards the edge, towards the cortex. You can see that in addition to becoming more fibrosed, though it's not visible on your screen here, an increase in fat deposition, one can definitely see that the number of lymphocytes has decreased markedly, though the thymus will continue to produce T cells throughout one's lifespan. It's just not as probably as active as it is in the juvenile and the later adult form of thymus. But easily recognizable by the appearance of these Hassel's corpuscles and a lighter staining medulla and an outer cortex. The features are still there, but a little bit more obscure.